Hi, I'm Mickey Willis, and this is Broken Truth.
Intro.
Okay, brother.
Let's do push-ups.
Tell me how many microchips are in my back.
Drinking
Lower.
frog, gay.
Lower.
Hi, I'm John Davidson. This is Broken Truth, and today I am with the lovely and talented Kevin McKernan.
What a lovely intro you just gave me. Thank you.
Thanks, buddy.
It's
How you
good seeing you again, by the way. Yeah,
You
in person this time.
I know, right? It's even better. Kevin is a fantastic researcher. You've done a lot of important things in this vaccine injury weapon world, whatever you want to call it. And what's new with you, Kevin?
Oh, yeah. I guess I wouldn't classify myself that way. In that it's somewhat accidental. I don't want to be here.
I don't either. But here we are.
But here we are. Yes, yeah. So we do tend to sequence and do genomics on nutraceuticals like cannabis and psilocybin, and a lot of these supplements people are taking. Everyone wants to get them tested to make sure they're clean of E. coli and salmonella and all those things, right? That's easy.
Sure.
But the moment we apply that to an injectable like a vaccine, we found things that we would never find inside of a nutraceutical. So it does make you question what the FDA's purpose is if the nutraceutical market seems to be cleaner than everything they regulate.
Right.
But lately, I'm sure you just heard from Jess. She's kind of gone over this new paper we put out, and that has the whole pub smear mob descending on it, which is kind of hilarity in motion.
Right.
But we're also looking at another angle on this problem, in that the FDA keeps coming back saying this DNA is non-functional and who cares about it? We can have some DNA in all these injections. It's fine. And I think the important thing to pay attention to is, now that they're removing the adjuvants from the vaccines, they're going to turn to DNA as their adjuvant. In fact, they've already done this in the Hep B vaccine. You'll see there's a cpDNA inside that vaccine.
So now for our viewers, an adjuvant is?
An adjuvant is anything that's poisonous that gets discovered after the fact. They reclassify it as an adjuvant.
Okay.
I mean, I'm being sarcastic, but basically anything that they find in vaccines they don't like, they will ultimately reclassify as an adjuvant, and saying it is something that helps stimulate the immune response.
It's like aluminum.
Like aluminum or thimerosal, and basically, most things that are in there that people object to, they'll just say, well, that's an adjuvant. We need that to help stimulate the immune response. So they're going to do that with DNA as well. They're already doing it in some vaccines, but now that their adjuvant list is getting narrowed, like they're removing aluminum and mercury, great. I'm all for that. Those have particular, properties as adjuvants. What I worry about is if they replace it with DNA, there's more nefarious things you can do with DNA than you can with just a metal. For instance, you could encode the DNA to encode for like a virus, or a plasmid that encodes virus. Many of you may know the entire COVID virus was at one point encoded in DNA. Like there were these clones that they made that Barrick and these people would ship around that had the entire, COVID vaccine, or I'm sorry, virus inside of a piece of DNA. So DNA can be a very versatile and dangerous adjuvant because of what people can sneak into it. They can sneak in self-replicating molecules. They can sneak in plasmids that replicate after they get in. So we're trying to draw some attention to that. So the thing with the FDA is they've been trying to claim this DNA contamination is non-functional. So we went looking through the plasmid literature to see that, what do people typically do that's different between the process 1 and the process 2 thing that happened. So this process 1 was done with PCR. PCR is a method that replicates DNA, but only replicates 4 of the bases. You've probably all been told there's A, T, C, and G, but there's another epigenetic layer of sequence information on DNA that's called methylated bases. So there's sometimes methyl C and methyl A. So it's really like 6 bases in DNA. Well, it turns out mammalian cells use that epigenetic language to sort out their own DNA from viral DNA. So viruses tend to methylate the A's, and we don't. Humans tend to methylate a CpG site, which is a C, then a G. It methylates that C. So when there's DNA floating around a cell that's got methyl A in it, the cells freak out, and they're like, that's viral or it's bacterial. We need to, marshal the troops and either kill ourselves or grow uncontrollably.
Trigger an immune response.
It triggers an immune response, an interferon response. So one of the key differences when I think Jessica walked you through how they changed this process from process 1 to process 2 with Pfizer. And one of the key things they changed is they stopped doing PCR and they started making plasmids, which are circular pieces of DNA that can self-replicate. Those DNAs, they grew inside of E. coli, which methylates it differently. It puts its kind of signature on it. It ended up putting methyl A, and it did this other methyl C that's different than human methyl C. It's a different pattern. Like humans methylate C when it's next to a G. Bacteria methylate C when it's next to an A or a T and 2 more GGs. It's called a DCM methylation site. The sequence pattern is CC. They call it a W, which is either A or T, and GG. So it has a, what is that, a 5 base pair sequence. When it sees that, there's an enzyme inside E. coli. It comes in and drops the methyl group on that C. So when that DNA comes out of manufacturing from Pfizer, it's now functionally different. And when you put it into a vaccine that gets injected into people with lipid nanoparticles, it triggers a pathway in the cells known as an interferon response. It's called CGAS-STING. It's a long acronym for stimulation of interferon genes, and that can lead to cancer. So we've been trying to sequence these vaccines to look at their methylation signatures to show the FDA that, look, it is not considered functionally the same. If it's differentially methylated, this DNA has a different impact when it gets inside of a cell. And here are all these plasmid manufacturing papers from 2010 that show it's a real crime to get this wrong. Like, if you get this wrong, you're going to have a much more severe interferon response. So those papers exist. One of them is called Karn et al., another one from Wang et al., another one from Lueck et al., and I'm going to murder the other name. I think it's Barbarello is the 5th one. But these papers walk through the impact of this methylation difference that's on the plasmid DNA that didn't exist in the PCR DNA. So we have a really strong case, the FDA, that you can no longer be calling this functionally equivalent because the plasmid manufacturing literature claims it's not. And Pfizer used the wrong strain. Like when they made this switch, they should have used an E. coli strain that was knocked out on these 2 genes that does the methylation. That's well understood art in the field. You just use an E. coli strain that's called a DAM- and a DCM- strain. So that doesn't methylate the DNA and it ends up looking a little bit more like the PCR DNA that they started the first process with. So that's been the latest work that we're doing, and we're going to run this through probably 10 more vials to just see if the methylation signals have ever changed. Because this would be if the methylation pattern changes, then we know they also switch cell lines.
Right.
And that's a big no-no. You can't be switching cell lines in manufacturing. And if they don't change, then we know they've used the same cell line for the entire time. That's fine, but there really shouldn't be any methyl signatures there, considering the trial was done on DNA that wasn't processed this way, and now they switched.
So you're essentially trying to prove to the FDA that this excess DNA or extra DNA is dangerous.
It's functionally different. They're claiming it's non-functional at all, and I have papers that demonstrate if you screw up this methylation pattern in manufacturing of plasmids for injectables and transfectable reagents, that is not considered functionally inert. So that's literally the sentence in these papers is this is not functionally inert. It is functional because you've methylated it. And we've got proof of that now with the Oxford nanopore sequencing that they've screwed up the methylation. They should not have used a strain that does this.
So is it possible that you inject this in a person and whatever the DNA is programmed to do just runs amok and your body doesn't know how to stop that? Is that what is?
So there's a couple problems. You're touching on another point that was in that paper. The other thing you can do with these Oxford nanopore systems, since they read single molecules and they're unlimited in the read length, we're getting 4,000 to 5,000 base pair reads sometimes off the vaccine. That tells us a lot of the DNA is fully intact and that DNA that Jessica spoke about didn't work. We could also see another step, one step earlier into their pipeline we can peer into with that data. We can see that they failed to actually linearize the DNA. So this gets to your concern over replication. Plasmids being circular means they have a unique capacity to replicate infinitely when they get into a mammalian host. Because it's circular, it works like a wheel. The polymerases that want to replicate this thing go round and round and round and round. And if it were linear, the DNA polymerases would get like one copy. You might get a little bit of amplification of the plasmid DNA when it's linear. But the moment you put it into a circle, that plasmid rolls like a wheel forever, and you get this massive amplification of the plasmid once it gets into a mammalian cell. So as a result of that, most of these people have protocols to cut the damn thing and linearize it so that it doesn't do that. And what we can see with this Oxford Hennefor data is they failed that step. It's probably maybe 60% linearized, 70% linearized from some of the runs that we've done. We can see about 20 to 30% of the reads are still, the cut site didn't cut appropriately. So that tells us that, okay, if they fail to linearize, there could be full-length circular replication-competent plasmids in some of the vials that when they get into patients, they expand and they keep growing with cell division. That would explain maybe some of the persistent stuff you're seeing at some of these conferences where people are reporting, well, this person has spike protein for 3 years, or this person has got his PCR positive.
Or maybe the clot situation.
Yeah, the clots as well is, there's a couple things going on there in that, so yes, persistence of the messenger RNA is probably a bad thing because that means your body is making more spike protein. And I think that spike protein itself is amyloidogenic, driving some of this fibrin clotting that's going on. So you don't want any persistence. You don't want the DNA or the RNA to stick around because I told you it was 48 hours, but the more it sticks around, the more spike it's likely going to make. The more spike persistence, the more clotting and amyloid mess you're going to get that Kevin McKernan is off studying. So yeah, I think that amyloid problem only gets worse knowing that there is a set of molecules that the patient can't clear that might be regenerating those spike proteins over time.
Okay.
So it's tied there. I don't know that the DNA itself is playing a role in those clots other than we can see it as part of the matrix in the clots. Like it's forming these neutrophil extracellular traps. So in the clotting process, sometimes circulating DNA is used as sort of a network for the clot to form on. And the clotting systems basically leverage circulating DNA to make a spider web of proteins. And we can see that process of mitosis occurring in 2 of the clots that Kevin McKernan sent us. It leaves a little bit of a signature in the DNA when it does this, because the DNA in that process is typically wrapped like a spool around a protein known as a histone. And that wrapping of DNA it likes to wrap in 11 base pair increments because that is kind of the persistence length that you can take a piece of DNA and turn it on itself. It takes about 10 and a half bases to get a full turn around a protein. So what you tend to see when you sequence nets is that you have an insert size of the DNA that oscillates like at a 10.5 base pair pitch because that's the number of wraps that you tend to get. So if it's wrapped, 3 times, it'd be 33 bases. If it's wrapped 100 times, it'll be, 1,100 bases. You can see that signature in the DNA that we got from Kevin. So there's definitely some signs of mitosis. Now, then the DNA that we got on those things was not the vaccine DNA. It was the patient's DNA. So the patient has a lot more circulating DNA in the blood when this is going on, probably because the vaccine created some epithelial scraping that sloughed off. That free circulating DNA was clean of any of the vaccine. I don't know when they were vaccinated, but what we could do in sequencing those patients' genomes is we could see they had some mutations that were in genes in that pathway related to fibrinolysis. This is the natural process of your body breaking down fibrin is impaired in some of those patients. It's N of 2, right? But we have to sequence more, right?
Now, is this DNA, is it SV40?
In the plasmid, yeah. In the case of Pfizer, yes, there's an SV40 component to it, and that's another whole rabbit hole on this mess. The SV40 side of this, encourage anyone here to, hopefully CHD will have Susan Humphrey's historical discussion of this, because this goes all the way back to the polio vaccine.
Right.
And she had some nice papers up there demonstrating that when you take the SV40 virus, which is about 5,000 letters long, and it is a double-stranded DNA virus, it ends up inducing tumors in a variety of mammalian hosts. Now, the epidemiologists like to butter this up and say we can't see it in the population. When we go look over the epidemiology since the polio vaccine came out, we don't see a cancer wave. There's nothing to see here, right? The problem with a lot of those observational studies is they're playing the same games they're playing now with COVID. It's shed, right?
Right.
So now you got the vaccine, I didn't. Let's track each of us for cancer. Well, if I've been sharing cigarettes and hanging out with you forever, then I got it too. and now I'm deemed unvaccinated. Therefore, you can't see a different differential cancer signal between us. So the shedding of the polio vaccine really screwed up the epidemiology for them to actually track cancer. And that really shouldn't be the metric for whether something is a carcinogen, as to whether you can, unleash it to the population. And let's see how, that's the wrong way to do it. What we do is we put it into animal models and see, does it create tumors there? With SV40, that's the case. All right, so that's the metric. The toxicological metric is not whether we can see it in the population, because those are incredibly confounded studies that are very hard to track across time. It's what happens with animal models. And in animal models, SV40 is certainly a carcinogen. Now, we don't have the whole virus inside the Pfizer vaccine. We just have some of the parts of it that we really wish they didn't use, because they're completely superfluous to their end goal. And exhibit A to this is Moderna. That Moderna doesn't have these parts, so they're proof that you don't need these parts. So they got lazy and left them in, or maybe that's being charitable. They intentionally left them in because they went and bought cancer companies right after this.
Oh, that's crazy.
Yeah, because
A crazy conspiracy.
Yes. Yeah.
But how convenient if you did accidentally release something that causes mass cancers.
And then bought
You have a cancer company.
Yeah. The interesting thing is they used the money from the COVID vaccine, the $43 billion, to buy the cancer company. So it fit well. But I don't know their intentions. It's just, you can watch their actions and judge for yourself. The components that are inside this vaccine, the SV40 components, there's some damning literature that implies that they could have known that this was going to cause cancer. In particular, that the sequence they left in there binds to P53, which is this tumor suppressor gene. There's a paper from Drayman et al. That shows that GC box binds to P53. You don't want anything binding that gene, because that gene needs to be busy cleaning up your genome, not Pfizer's mess.
It's a guardian of the genome.
Guardian of the genome, yeah. It's like throwing a bunch of decoys at the guardian of your genome, right? You don't need that. So that's a problem. Wafik-Eldieri's shown that the spike protein itself downregulates P53. So now we got two things that are messing with the guardian of the genome. That's a bad idea. Dean et al. Has shown that piece of DNA just traffics things right to the nucleus. So that brings on an integration risk. That's another way you can drive cancer. I actually think the bigger cancer risk is step one, which is once the DNA gets into the cell, it triggers this interferon response. That's the thing we should be looking at immediately, because that we know happens.
Sure.
Whether it integrates, well, we'll have to keep studying that. That's going to take us time. We're going to need IRBs and CLIA labs and genetic counselors to go into that rabbit hole. And that'll take years for researchers to resolve. But we already have got papers published now in Germany from Ulrich Kammerer, where they take the Pfizer vaccine, they put it onto ovarian cancer cells, and they watch them grow over time, and the SV40 sequence is staying along for the ride. So we know it gets into the cell, and we know it persists in the cell for a couple cell passages. So the cGAS-STING pathway we know is on. The other reason why we know that pathway is on is there are 5 papers that have published through peer review where they looked at vaccinated versus unvaccinated people, and they looked at their RNA, which is kind of like looking at, if DNA is your hard drive, RNA is like your task manager of all the programs that are running. They're going on and off, right? So it's much more of an ephemeral signal, but it shows you which genes are being turned on and off between the vaccinated and unvaccinated. So a few papers for the record here are Lee et al. Is one of them. Ryan et al. Is another one. Chakrabarty describes both of those. Odek et al. Is a fourth, and Nabel et al. Is a fifth, and even Krowski 6 is another paper. What these papers show is that if you look at the differential gene expression in the patients that have been vaccinated, we can see the cGAS-STING pathways getting aggravated. So and we can see the plasmid DNA in the patients. Now, we don't know if it's integrated, but we know it's in the blood supply, because these were people who got blood draws. And the process that they were using to sequence RNA doesn't do a very good job sequencing DNA, but we can still see it.
Yeah.
And so those are 5 peer-reviewed papers where they weren't even looking for this. We pulled their data out of NCBI, because when you publish a paper, you're supposed to put your sequence data public. You go to that database, you pull out their sequence, scan it for Pfizer plasmid sequence, and bang, 5 papers all have evidence of the sequence from the vaccine. And that's in the blood supply of patients. So it's not just like a problem in the vials. It's made its way into people, and we can detect it accidentally when they sequence people's bloodstreams, that this is there. And we can look at the RNA in those patients and see that it's only there, it's also aggravating their cGAS-STING pathway. So the precise expected outcome of flooding a cell with DNA like this, we can actually see the differential gene expression of the patients these people were studying.
Wow, Kevin, this is really fascinating stuff. Maybe a little scary.
Yeah,
What's the silver lining? Are there ways to treat
Yeah, I feel like that naked guy running around Times Square saying the world's ending. That I'm not typically in this position. I'm in the position of sequencing and helping people build their own medicines, nutraceuticals. I'm a big proponent of getting outside of the FDA system because it's centralized and everyone can see it has no accountability. So what does that mean? That means we need to turn to natural products. We need to turn to supplements. We need to turn to generic drugs, like a lot of the things you're seeing these physicians turn to now that they can't trust the FDA. And I've been really excited about building tools that help decentralize that and make that, how do we self-regulate knowing we don't have a regulator we can trust?
Right.
And that free market approach, I think, is a better approach. So the silver lining here may be that, okay, more people have woken up to the fact that they can't trust the FDA to regulate their medicine. And they're going to have to start thinking about getting outside of the healthcare system, maybe using direct primary care physicians, these aren't physicians who are captured, who say safe and effective and get 70 vaccines, right? These are physicians that aren't captured by insurance. You have to pay out of pocket, yes. There's other types of crowdfunding, like health insurance bodies that are forming. I think crowdhealth.com is one I've been looking into. I hear a lot about them in the crypto space. A lot of those folks are using that. That's a bit of like a community pooling organization where it's more like a co-op for healthcare. It tends to be a lot cheaper than traditional healthcare.
Okay.
And then I like to turn people toward medicines you can grow in your backyard. Whether that's cannabis, fungi. There's a host of other medicinal plants out there, from dandelion to you name them. You can see them here in the hallways that people are turning toward, where we can learn to actually grow those and QC those and get some quality statistics around those tools, and get around this concern over snake oil. The whole FDA was formed over the fear of snake oil, and now we've come to learn that they're the snake. And
Well, I mean, the caduceus is sort of a hint.
yes, it was nice.
So, Kevin, how can people follow you and keep up with you?
I'm on Twitter and on Substack. And if you want to learn more about decentralizing medicine, we do run a conference every year called CanMed. It's a lot of physicians just like this. I'm really intrigued by this. I feel like it's a little bit of we have echo chambers at a building here. This is a great group of physicians that have figured out what's going on with COVID. The CanMed group these people have been persecuted for 50 years trying to get cannabis to kids and to cancer patients and epilepsy patients, right? They have been through this war for decades longer. It's just a different war. But it's the same battle. It's the same battle for medical freedom. And so you can find me there. We run that conference. It's going to be at Lake Tahoe in, I think, June 12th to June 14th. But those physicians have lots of war stories about what we're about to go through here. We're now at this stage where ivermectin is going to go over the counter. And the first thing they're going to run into, because we saw this in the cannabis field, is who's going to make it and who's going to QC it. And in Texas, that's the problem right now.
Right.
Someone's going to make it, someone's got to QC it. So they need to build a network of analytical chemistry labs like we have in the cannabis market to go and QC all of the products that they want to make and bring over the counter. And we want ivermectin to spread as fast as cannabis spread from a legalization standpoint. I think the main irony is that it is spreading in all the red states and cannabis is spreading in all the blue states. And there's got to be a way to make the medical.
Maybe there's a chocolate peanut butter combination that we could.
We should mix a little ivermectin with some CBD and see what happens.
I'll participate in this experiment.
Yes,
It's for science and humanity.
it's it. It might lower your fever too.
Kevin, thank you so much.
Excellent, Tom. Thank you.
Thank you.
I believe in the Constitution. I believe that one case, the right case, can change the world. If you believe this too, Freedom Council is for you. Pursuing a major case can be overwhelming, but we provide a community of practice so that you have the resources available to support your case. My name is Warner Mendenhall, and we are Freedom Council. Join Freedom Council today. freedomcouncil.org.